Okay, my name's Lucy Davison. It's my pleasure to chair this session on surveillance. We've got a slight change in running order thanks to our fantastic lunch that took slightly longer than anticipated. What we thought we'd do, because we have a hybrid remote speaker, Alan Radford, who's joining us from Liverpool, will go with his talk first, and then we'll revert to Dr. Helen Roberts and then Professor Darby. So we will do the same talks, just in a slightly different order, and we'll try and stick to 30 minutes per speaker and allow us to have some time for discussion later on. So it's a real pleasure to introduce Professor Alan Radford from the University of Liverpool, who's going to be joining us remotely to talk about SAVSNET, the Small Animal Veterinary Surveillance Workshop.
You're listening to me today, but we are very much a team, a big team, but the three people who run SAVSNET are myself, PJ Noble, and Gina Pinchbeck. PJ is a clinician, but also a data scientist and has a big interest now in artificial intelligence. And Gina Pinchbeck is an epidemiologist. My background is in veterinary virology. So what SAVSNET does is it reuses existing information, in this case, electronic health records, for animal disease research and surveillance. I'll give you a little summary of how we do that, and then I'll talk about the work we've done on canine enteric coronavirus. So a busy slide, but it's quite straightforward. We collect data from two places. We collect data from, and I'm hoping you can see my mouse. We collect data from veterinary practices here at the bottom left, and we collect data from commercial diagnostic laboratories. In both places, information is recorded on a computer. And the question we asked ourselves was, could we use this information, reuse this information for surveillance and research? The information that we collect is very variable, very messy, some people might say. It's very big, so 5,000 records a day from vets in practice, tens of thousands of records a day from diagnostic laboratories. We bring that data to Liverpool. The first job that we have to do is to clean that data. We call it map the data and make it research ready. And increasingly, that is happening now in real time. Obviously, we can't read these records. We can't read all of these records. There's too many. There has to be a data solution to identifying information in these records. We can add data, which we're increasingly doing. The best examples are deprivation data and climate data. And then we use the data for lots of reasons. You don't need to look at these particular pictures, but we use them for surveillance. We use them for research. I think there's a big overlap between those two things, research and surveillance. Some people think they should be completely different. So let's have a little look at the laboratory data. So this is the process. A vet in practice takes a sample. They send the sample to a commercial diagnostic laboratory where a test is performed, and the result goes to the vet. What we do is we also collect the result, and we can use that increasingly in real time to look at the distribution of pathogens in the UK. And hopefully you can see a little bit more of this slide than I can. There should be a URL at the bottom of this image. I'm not going to mess. Oh, there we go. You can go to this URL and see some examples of real-time laboratory data, but it shows us where viruses and bacteria are being diagnosed and when. The other thing that we are increasingly doing, because we have a genomics expertise at Liverpool, and you'll hear from Professor Darby later on today, is we want to sequence things, not just get the result, but we want the sample. The laboratories throw the sample in the bin. The laboratories throw the sample in the bin. So maybe could we collect some of those, not all of them, there's too many, and bring them to Liverpool for enhanced phenotypic and genotypic testing. And we've done that. We call it the virtual biobank. The virtual biobank. This picture here shows our first go at the virtual biobank with canine parvovirus, but we also used it to great effect to do some COVID serology in dogs and cats during the pandemic phase of COVID. So that's the data from laboratories. The data from vets in practice is quite different. It's what we call an electronic health record. It has information such as the date of the consultation, the postcode where the owner lived, the species and breed and sex, the vaccination status. It also has the free text. This is a wonderfully complex piece of textual information that has within it a lot of important data, but not in a structured way. But it turns out with modern computing and data science that you can identify important features in free text quickly enough and efficiently enough to be useful. I'll show you an example on the next slide. This is roughly where we get our data from these practices. So here's a free text. Removed tick from head. Frontline 6 weekly, explained help for 4 weeks. It's not written very nicely because vets are busy and quick and they don't check it for spelling mistakes, but it says there was a tick on an animal on this date, and we have the postcode of where the animal lives. And we wondered whether we could use that information across all 10 million records that we have to look at the temporal and spatial pattern of ticks in the UK. And we have now published 2 papers on this. This one descriptively showing the pattern of these tick consults and showing for the first time, I think big data is really good at this sort of thing. It shows for the first time that the pattern in dogs in blue is different to the pattern in cats, which is shown in orange. The cats peak earlier in the year, and they also have this second autumnal peak. We've since gone on to model the risk across the UK, and so we can even predict places where ticks will be active and at what time of the year, even though we don't have actual data from those regions. And we do that based on things like, because we know the land coverage of that particular region of the UK, or the temperature at that time of year, or the humidity, or the elevation. And this work was just published in 2024. This is a really nice example, and it reminds me to say that SAVSNET data is available for other people. It's not data for Liverpool alone. These researchers were based, the lead research was based in France, and it's a really great example of how we can make data available for fantastic scientists to do good research. So maybe you might apply for access to SAVSNET data after hearing this talk. One of the things we've done a lot on is around antibiotic use. And I'm not going to spend much time on this slide, but this shows that some practices use a lot of antibiotics compared to others. This one here shows that in both dogs and in cats, antibiotic use in the UK has been falling. And this one, which we're particularly proud of and was led by Gina Pinchbeck, the reference is here, shows that if you talk to these vets, the high users, and show them that they are a high user, then their antibiotic use falls. And that's what's shown in this picture here, this figure here. So that's how SAVSNET works. What about coronaviruses? Well, rewind the clock to February the 6th, 2020. And in the UK, well, what was happening? COVID, this is COVID. So we're about here, 6th of February. This was just before COVID was really starting to increase in the UK. COVID was on the news every night, but the cases were still fairly small, 6th of February. But in the local newspapers and online, there was a lot of talk about a dog vomiting virus. This seemed to be unusual because it was very infectious, and the affected dogs seemed to be very severely affected. This wasn't 1 or 2 vomiting episodes, it was 8, 9, 10 vomiting episodes. Dogs were being hospitalized. Dogs were not dying, thankfully, but it was much more severe than any pattern of disease that had previously been seen. So we obviously got a little bit interested in what was going on. Maybe it was COVID. We didn't know. So there's a lot of information on this slide, but what we were able to show with our data from vets in practice was that there was indeed a very unusual pattern of gastroenteric disease in dogs. That's what these red dots show here. Much higher than we had in previous years. Gastroenteric disease was gently seasonal until January 2020, when it became, the levels were much higher. That signal was not present for respiratory disease in dogs. It was not present for gastroenteric disease in cats. It was present for meropitant. Meropitant, as many of you will know, is a drug used to treat vomiting. And meropitant use in dogs was very high. Meropitant use in cats was stable. And this was another way, this was a text mining approach like we used for ticks to identify vomiting dogs. Vomiting was very high in dogs. It was all over the UK. That's what the veterinary data showed us. We did some epidemiology to show that, led by Gina Pinchbeck, to show, and a case control study to show that this was likely to be infectious, and importantly, that humans did not seem to be affected, nor was there any link to COVID that we could see. So that was important field epidemiology data. When we looked in the laboratories, parvovirus was perhaps what most people were thinking. These data here show parvovirus over time in the diagnostic laboratories. These are the number of samples being tested. And this is the proportion testing positive. And to us, that looked fairly flat. But when we looked at canine enteric coronavirus, the proportion testing positive was very seasonal. It wasn't something we had seen before. And the spike was in January. So we thought maybe we should test for some of the samples from our case control study and some samples from the virtual biobank and sequence them and see what they are. So this is M gene sequencing. What that showed was that most of our positive cases were infected with the same virus. And when we picked some of those for whole genome sequencing, again, the virus was identical. I think we sequenced 5 in this first season. And this virus was spread all over the UK. That's what this shows. The red arrow, dog 7, we'll come back to in a moment. So remember this one. Dog 7 is an example of what we call the major variants from 2020. Well, you know about canine enteric coronaviruses, but very quickly, it's an alpha coronavirus. It's closely related to feline and porcine coronaviruses. There have been recent variants discovered in humans, sporadically, rarely, which raises an interesting new angle, perhaps. And historically, there have been 2 presentations: mild diarrhea, very mild, I could have written, possibly inapparent in many cases, mild diarrhea, or sporadically severe infections in puppies, often with co-infection. And these infections have often coined this term pantropic canine enteric coronavirus. We could discuss that term later, maybe. So roll on to 2022, and essentially the same thing was happening. Mystery dog vomiting virus spike in Yorkshire. So we ran again the process. We looked through and said, if coronavirus, canine enteric coronavirus, is again causing a spike in disease in dogs, how do those viruses relate to the ones from 2020? That's really what we wanted to know. So again, we had 46 samples from vets. It was actually quite difficult getting samples from vets in 2022. I think they were tired of COVID and very busy. But we got 46 samples and 18 tested positive. We got 87 samples from the virtual biobank, 27 tested positive. And with Al Darby and colleagues, we went ahead and sequenced them. So a busy slide, but it's actually quite straightforward. So here's the phylogeny. This is the M gene phylogeny. The 2022 samples are shown in this line here, in the purple, and this line here. The major variants, so this was the virus that we found most commonly, is shown here. This is the major variant from 2022. And then we found quite a lot of other variation. In 2020, we found a major variant and very little other variation. In 2022, we found a main variant, but quite a lot of additional variation. And when we compared that to the viruses from 2020, it wasn't the same virus. It's important to say. This just shows the distribution. The dark purple is the major variant. And again, it was all over the UK. All over the UK. When we whole genome sequenced, what we found was recombination. And so this is comparing the, I'll just go back, sorry. This is comparing this virus, 1022, which is an example of the main variant from 2022. So this figure is a similarity plot comparing that virus to the major variant from 2020, a minor variant from 2020, and a published sequence called A76, which we chose because it was the closest in a region of clear recombination. This identifies the 5' end of spike as a clear recombination in this major variant from 2020. On the right here is the whole genome phylogeny for the core genome. So when we take out the recombinant regions and just look at what's left of the genome, we find that again there's this major variant and also some minor variants too. Our sense from the phylogeny was that in order to understand how these viruses are evolving, you need both things. You need to look at the spike, absolutely, but you also need to look at the core phylogeny, what is left after you remove the regions of recombination. We could talk a little bit about, if you want to talk about the methodology that we used CISPR and amplicon tiling and also nanopore, increasingly perhaps nanopore. Obviously, there's been discussions about human viruses, and our viruses were different to those in the important 5' regions of the spike virus. So we felt there was no risk to humans from the viruses that we were seeing. How am I doing on time? I don't actually know what time we started. I should have set a timer, but I've got 2 or 3 slides to go. One of the things I thought you might be interested about SAVSNET is because everyone's talking about artificial intelligence now and things like ChatGPT. This is work led by my colleague PJ Noble, and it turns out that you can get the computer now to spot this outbreak as well, in fact, better than our previous approaches, which relied on what the vet was writing in the free text. So this is based on topic modelling, and this is based on what the vets were writing in the free text and how we were searching for patterns of words. This process of topic modelling is very powerful. It identifies groups of text that contain words of a particular relationship, and the words here in these outbreak consultations were diarrhea, vomiting, feces, eaten, vomited. This was clearly something to do with gastroenteric disease, and it required almost zero work from a human. So AI, we think, has great potential for outbreak detection. That's what this slide talks about. If you want to know more about our work on artificial intelligence, which is led by PJ Noble, then please do check out this website. There's a lot of stuff coming out from PJ at the moment. In terms of future work, if you're interested, we have completed a longitudinal survey of canine enteric coronavirus diversity over a whole year now. Clearly, so far, the work I showed you was sampling in 2020 in the outbreak, sampling in 2022 in the outbreak. And what we wanted to know was what was going on in between these winter peaks. So we sampled for a whole year from the virtual biobank. I'll leave that slide there. In terms of conclusions, I think we found a new pattern of seasonal winter gastroenteric disease in the UK. I think it's severe in some affected dogs. I don't think last year was as bad as previous years, which all sounds like COVID to me. Lots we could talk about there. We use the word associated with. We don't know if it's caused by, but it's associated with recombinant CE-CoV variants. No known zoonotic risk. We've shown that if you can bring together vets, owners, diagnostic laboratories and universities, you can very efficiently carry out epidemiological, microbiological and genomic diagnosis in the face of an outbreak. Fake news I thought you might find amusing. Our local newspaper in Liverpool did an interview of a Liverpool professor who will remain anonymous. It was me. And I talked over and over about this is not COVID. This is not COVID. Yes, it's a coronavirus. It's not COVID. And this was the headline that they produced. Interesting. How this virus relates to what's going on in Cyprus, I don't think we've had that conversation, but I think there are a lot of similarities. I'm going to finish on this slide, really just to bring you back to SAVSNET and to make the point that SAVSNET collects data on all the reasons animals go to the vet, and so you can use it for lots and lots of things. I've shown you just a few examples focusing on canine enteric coronavirus. We now have projects on equine surveillance led by Gina and farm animal surveillance led by me at the moment. And we have a sister project in University of Minnesota This makes the point that we think our science could work in lots of other places. It's not complicated. Collect data, analyze it. And I'll leave you with a big list of collaborators that I won't go through. This Liverpool is one small part, an important part, but one small part of a very big network. And I'm massively grateful to, I will identify a few, our practices, in particular CVS, our laboratories, in particular nationwide, and IDEX, and also our funders, originally BSAVA, BBSRC, and our major funder currently is the Dogs Trust. Thank you very much.
Thank you very much indeed. Perfectly timed and a really fascinating, really inspiring talk. Does anyone have any questions? I've got lots, but I'll let the audience ask first. We've got a few minutes for questions.
Very interesting talk. I'm quite fascinated by the recombination event, which happens to be in the spike region. Are there documented other sort of recombination points in coronaviruses, particularly non-structural proteins?
So I think there will be people talking today who can answer your question much better than me, but I will say my little bit. So here again is the recombination that we observed, and we all focus on this region here. Our eyes are drawn to the spike. It's just what everyone focuses on, but clearly there is recombination happening potential recombination happening in other places. I think there is a hotspot for recombination in the spike, but other than that, we've not looked for particular regions that are more likely to recombine in canine enteric coronavirus. I think when we've analysed the whole genome sequences for the viruses we've collected over a whole year, then I think that will be really interesting, because what I hope is we'll see this virus and how it changes over the following summer. Because my belief is that these spikes will only occur if something happens to make a new variant that the dog population in the UK or a particular region of the UK more susceptible to it. So I don't think I've really answered your question. I think there are people coming who will be able to talk much more about sites of recombination in coronaviruses. But it's a great question and something we need to understand, because clearly recombination is massively important to what these viruses are doing.
Can I just ask also whether that includes the receptor binding site or the furin cleavage site?
I think it does include the receptor binding site, but if you can email me, I will probably be able to be much more definite about an answer to that.
Thank you.
There's one question at the back. Thank you.
Sorry, can I answer that question, Peter?
Oh.
Hey, Alan, lovely talk. It's Julian. I'm just going to answer that question.
Thank you.
Yeah, so recombination can occur anywhere, but when you see it, it's because that viral genome survives that recombination event. And there are particular sites in spike where tolerance of genomic changes can occur. So you can either get insertions or deletions, but it will occur throughout the genome.
Thank you. Are there any more questions? Yeah. I think we have one more.
Hello, thank you. I was just wondering, when you're collecting data on such a massive scale, how do you control for confounding variables? And what are the kinds of things that you're looking for to say that your results are true versus coincidental?
Yeah, it's a great question. Obviously, we are not collecting data in any controlled way. We absolutely recognise our data has within it many biases, probably, some of which we know about and some of which we almost definitely don't. I think if you, one of the typical studies we will do is a case control study. So we compare animals with a particular disease to animals without. That's most of the publications we've done on individual diseases like ticks or fly strike or epilepsy, for example. So we do case control studies a lot. I think with things like if I just show, I'll tell you what I'll do if we've got a tiny bit of time. This is our website. If you go to real-time data, you can have a look at something like this, the main presenting complaint. So this is live data that everybody can look at. It's public. And I think it's the fact that we've had data now for many years. So this is looking at, if we look at dogs and gastroenteric disease, I hope this works. Hope it works. There we go. So this is dogs and gastroenteric disease. And you can see, I mean, there's no statistics here, but you can see that the signal is reasonably stable and then goes crazy. This spike we think is due to COVID because vets are not going to, animals are not going to the vets anymore. And then we see this regular seasonality for two more spikes. So I think we get more confidence where we've got a long period of data. And I think the only other thing I would say is that we try not to rely on a single piece of evidence. So that's why I think here it was really important to us. I hope you can see my slide still. It was really important to us to not just look at gastroenteric disease, but to check other signals like respiratory disease, like neuropatents. This one's text mining, to look in the laboratory data. And when we have data from multiple different places saying the same thing, it gives us much more confidence. Of course, we can't still say that canine enteric coronavirus, we cannot say canine enteric coronavirus causes this spike. And that's why I really still emphasize what I did on the summary slide, which is we use the word associated in order to show that this virus, these viral variants cause severe vomiting and diarrhea. We'd have to do experimental infections. I hope that answers it goes some way to answering your question.
Yes, it does. Thank you very much.
Thank you. I think we will have to move on, but that was a really great talk and discussion. Thank you very much again, Professor Radford.